Hello everyone, welcome to the ITLS Academy. Today we are going to discuss another topic that is your lactic acid fermentation. Okay, so before starting the lecture, I just want to tell you what does this fermentation means or what is this lactic acid. So first of all, I am going to tell you the definition of the fermentation. Okay, so basically the fermentation is what? Do you know? Any guess? Okay, now I'm going to tell you fermentation is what? Fermentation is a process in which substances are break down into a simple substances. Complex substances are break down into a simple substances. Basic. It is the basic definition. Okay. Now here the microorganisms are used during the fermentation. What should we do? We use our microorganisms. Okay, and these microorganisms are either they are bacteria or either we are use yeast for the fermentation. Okay, so fermentation is what? Fermentation is a process in which suppose that what happened here, a breakdown of the glucose take place. Okay, and this breakdown of the glucose will take place in the presence of bacteria or yeast. This is called your fermentation. Okay, so fermentation are used in a different industry like your manufacturing of wine, manufacturing of wine, beer. Okay, now what is this lactic acid? This is the this is about the fermentation. Now the second thing is your what is lactic acid? So lactic acid are defined as simply it is defined as a chemical which is produced by by the body when the cell break down carbohydrate for the energy. Okay, so simply it is a compound which is produced due to the breakdown of the carbohydrate. Okay, this is called what? This is called your lactic acid. So in today's lecture we are going to discuss about what is this lactic acid fermentation. So let's start today's lecture. Lactic acid fermentation is basically is a metabolic process by which glucose or other 6 carbon sugar, that is your disaccharide of 6 carbon sugar, that is your sucrose or lactose, it will convert it into a cellular energy and the metabolite lactate. Okay, so lactic acid fermentation is what? Lactic acid fermentation is basically a metabolic process. It is clear that it is which type of process? It is a metabolic process. Okay, so metabolic process in which either glucose or other 6 carbon sugar, that is your either disaccharide or 6 carbon sugar, that is your sucrose or lactose, converted into a cellular energy and the metabolite lactate, which this lactate produces lactic acid in a solution. Okay, and basically it is an anaerobic fermentation reaction, means that this reaction will be take place in the absence of oxygen that occur in some bacteria and animal cells such as muscles cell. Okay, basically this is what? This is your lactic acid fermentation. Now the next is your lactate dehydro lactate dehydrogenase. It is what? It is a catalyzes the interconversion of pyruvate and lactate. Okay, see, lactate dehydrogenase is what? They catalyze the reaction. And which type of reaction? Interconversion of the pyruvate and lactate with concomitant interconversion of NADH and NAD plus. In homolactic fermentation, one molecule of glucose is ultimately converted to a two molecule of lactic acid. Okay, so if we talk about the homolactic fermentation, in this process, there are one molecule of glucose which converted into a two molecule of lactic acid. Now, I'm going to tell you what is this homolactic fermentation. Okay, so homolactic fermentation is basically defined as a process that converted sugar into a cellular energy. Sugar into a cellular energy and lactic acid metabolic by product. Basically, it is what? It is a homolactic fermentation. Okay, and in this process, there is a one molecule of glucose. It is converted into a two molecules of lactic acid. Heterolactic fermentation, in contrast, yield carbon dioxide and ethanol in addition to lactic acid in a process, and this process is called a phosphocatalysis pathway. Okay, so there is homolactic fermentation and there is a heterolactic fermentation. In homolactic fermentation, it is clear that one glucose molecule is converted into a two molecules of lactic acid. But in heterolactic fermentation, there is a carbon dioxide and ethanol are also produced along with this lactic acid, and this process is called a phosphoketolysis pathway. So this is all about the homolactic and heterolactic fermentation. Now the next is now the history of this lactic acid fermentation. There is a French chemist, Joseph Louis Gay-Lussac, who has who was especially interested in a fermentation process. Okay, Joseph Louis Gay-Lussac, he was a French chemist and he was especially interested in a fermentation process, and he passed this fascination to one of his best student, Just von Liebig. With a difference of some years, each of them described together with a colleague that the chemical structure of this lactic acid molecule that is known today, they had a purely chemical understanding of the fermentation process, which mean that you cannot see it using with the microscope. Okay, now in 1857, the French chemist Louis Pasteur, he is the first who described the lactic acid as the product of microbial fermentation. Okay, according to Louis Pasteur, he described that this lactic acid is the byproduct of, is the product of the microbial fermentation in 1857. Now, types of pathway. One is your homofermentative and another one is your heterofermentative. In homofermentative, this glucose is converted into a 1,6-bis fructose. Okay, glucose is converted into a 1,6-bis phosphorus, and in this process there is a 2 ATP which is converted into a 2 ADP. Then this 1,6 bifosfructose is converted into a 2 glyceraldehyde 3 phosphate, and this 2 glyceraldehyde 3 phosphate will be converted into a 2 pyruvate. Okay, and during this conversion 2 NAD is converted into a 2 NADH, and 4 ADP is converted into a 4 ATP. Now this pyruvate are converted into the lactate. That's how this glucose are converted into the lactate. Now in heterofermentative process, where along with the lactic acid, ethanol is also a product. So in this process, first of all, the glucose is converted into a 6-phosphogluconate. Okay, here again this ATP is converted into a ADP, and NAD is converted into a NAD2H. Now here this 6-phosphogluconate are converted into a pentose phosphate, and here a carbon dioxide is released. Now this pentose phosphate are converted into a glyceraldehyde 3 phosphate, and this glyceraldehyde 3 phosphate is converted into a pyruvate. Okay, and here this pentose phosphate is there is a coenzyme react, and it will convert it into a acetylyl co, and this it will convert it into a acetaldehyde, and this will convert it into a ethanol. And here in this process again NAD is converted into a NAD2H, and this pyruvate is converted into a lactate. Okay, so this is a two pathway of the conversion of the glucose or the fermentation. One is your homofermentative and another is your heterofermentative. Now the next is your lactic acid bacteria. Okay, so phylum Firmicutes, class Bacilli, order Lactobacillus, genera Lactococcus, Lactobacillus, Streptococcus, Leuconostoc, Oinococcus, Vagococcus, Pediococcus, Eriococcus, Carnobacterium, Enterococcus, Tetragenococcus, Visalia. Okay, catalyst test if performed, then it will be a negative. Devoid of the cytochromes and porphyrins, microaerophilic, and it will be grow at the range of temperature 5 to 45 degree Celsius. Okay, that's why it will come under the danger zone. Now the next is your types of the lactic acid bacteria. That is, the first one is your homofermentative process, and the second one is your heterofermentative process. Okay, so this homofermentative process, this is your called a single fermentation product in which lactic acid via glycolytic pathway. Okay, only one product is formed, that is your lactic acid. Okay, and that's why it is called a single fermentation product. Here you see that it is a glucose, it will convert it into a lactic acid in the presence of either lactobacillus acidophilus and lactobacillus casei. Now next is your heterofermentative. In heterofermentative, there is a byproduct or also produced along with this lactic acid. Okay, and this byproduct are what? Either ethanol, acetate, and carbon dioxide that we already see in the cycle which we discussed in the earlier. Okay, via 6-phosphoglucanone gluconate pathway. Here you see that here is a glucose. It will convert it into a lactic acid. Along with the lactic acid, we also get a two byproduct. One is your ethanol and one is your carbon dioxide. And this reaction will be take place in the presence of Lactobacillus mesenteroides. Okay. So this is the types of the lactic acid bacteria, homofermentative and heterofermentative. Now the next is your process of lactic acid fermentation. One is your natural and another is your control. Okay, so this lactic acid fermentation process, either it will be done naturally or either it will be done by a control. So in natural fermentation, what should we do? We isolate the natural flora, and this natural flora will be grow during the fermentation process. Okay, suppose that we have a natural flora of different microorganism. Now what should we do? We isolate it. Now after isolation, this will grow, and this is called your natural process. Now raw material have a high microbial loads. Either they contain a gram negative bacteria, either they contain a gram positive bacteria, or either they contain a yeast and mold. Now this process can be enriched by the addition of salt or by a certain protein ingredients. Okay, so this process is either enriched by when we added the salt or when we added a certain protein as an ingredient, and this addition of the salt and the addition of the protein will promote the growth of the lactic acid bacteria. LAB is what? It is a lactic acid bacteria. Okay, so this growth is either controlled by the addition of the salt or addition of the protein. Now the next is your control fermentation. Okay, this control fermentation is involved. What should we do? We have a natural flora. We all know that. Natural flora. Now we pasteurize this natural flora. Okay. In pasteurization, what should we do? The pathogens will be removed or killed. Now, after that, what should we do? We added a starter culture of autocotonos or allocotonos. This autocotonos is a starter culture which are isolated from a reused on the same raw matrix, and allocotonos is a starter culture which are isolated from a certain raw matrices, but it is used to fermentate the various product. Okay, now this quality is controlled by a industrialization of fermentation process. Okay, this quality control of this control fermentation is essential for the industrialization during the fermentation process. So what should we do? We remove the natural flora by our pasteurization. Then we add a starter culture, either autochthonous or allochthonous. Okay, in autochthonous, what should we do? We added isolate. We uses the starter which are starter starter culture which are already isolate from a same raw matrix, and this allo cotonas are the starter culture which are isolate from a certain raw matrices. Then their quality should be essential during the fermentation process which are done in a industrial purpose. Now there are some advantages of this controlled fermentation. Okay, they are pure starter culture which we added. Okay, now there is a no risk of the contamination as a pasteurization step is done. We already performed the pasteurization. When we perform the pasteurization, we already remove the pathogen microorganisms. Pathogens, microorganism. Now, when these microorganisms are removed, then there is a no risk of the cross contamination. Okay, and one more thing, what should we do? We added a pure starter culture. It also enhanced the organolactic properties due to use of the functional starter culture. Okay, it increased the fermentation efficiency. This controlled fermentation also increased the fermentation efficiency. Now the next is your chemical synthesis. It is used to produce the racemic mixture of lactide is hydrolyzed by a concentration sulfuric acid so that we get a lactic acid along with a ammonia salt. The next is your microbial synthesis, and this microbial synthesis is used as a pure L plus and D negative, and it will produces a lactic acid, mainly a carbohydrate, which is a substrate of a lacto mm, a lactoacidic acid bacteria. Lactic acid, sorry, lactic acid bacteria that at rhizosome ori orezi. Okay, so this is about the chemical synthesis of the lactic acid and microbial synthesis of the lactic acid bacteria. Now the next is your lactic acid fermentation microbial. Okay, inoculum should be take place at the range around 5 to 10% of the liquid volume of the fermented, and the temperature should be at the range of 35 to 46 degrees Celsius, and pH should be at the range of 5 to 6.5, and here it is maintained either by adding the ammonium hydroxide. Okay, and this is a batch fermentation process. Here a batch and fed batch fermentation, high concentration of the lactic acid is achieved, and this process is a continuous process. High productivity of the lactic acid is achieved. Airlift bioreactor is preferred for our rhizopy, orzi, and filamentous fungi. This is your plant of the fermentation. Here, this is your fermenter. Just a minute, I will change the ink. This is your fermenter. Here we added the sugar, we added the alkali, we added the nutrients. Okay, here this is your separation, this is your cell cycle, this is your nutrient cycle. Here it is a purification, then recovery. Here a pure lactic acid we get. So this is your simple diagram for the fermentation process. Fermentation process. Okay, here the purification is done. Then after purification, what happened? Recovery column. Then this recovery column is going to a cellular unit. Then after cellular unit, what we get are, we get a pure lactic acid. Simply. Now the next is your fermentation product. raw material and the product. If we take a raw material like milk, then we get a fermented product like your yogurt, buttermilk, not a fermentation product, or you can also set that fermented product. Okay. If we have a raw material like vegetable, then we get a product like sauerkraut, kanji, pocho, pickles. And if we have a raw material like fruit, then we get a product like your fermented olive, sweet cherry, and capper berries. Now the next is your kanji. Kanji is what? Kanji is a fermented black carrot beverage. Okay, what should we do? We all know that there is a black carrot. So this black carrot are converted into a beverages. Okay, and this beverages will be fermented. Then this is your kanji. This kanji are rich in beta carotene, anthocyanin, and antioxidant. And it will also acquire a diuretic, smoothing, hepatoprotectic, and uterine stimulative property. And it is mainly consumed in different part of the India, like northern India and Pakistan. Okay. Now the next is your procurement of the this fresh black carrot. How we form this kanji? How this kanji will be prepared? Now, what should we do? First of all, we have a black carrot. We procurement. Then wash it with a KMC, that is your potassium metabisulfite solution, and the percentage of this KMC will be 0.01%. Then after that, we peel the carrot and shred it into a equal size. Then this carrot pieces are fermented. Okay. Then we give a heat treatment about 70 to 80% of the temperature for a 15 minute, and along with it we are also use a common salt and dry about 1.5%. Then we add 1% of grown culture of lactobacillus for 20, and this culture grown at least for 24 hours. Then we boil it, we cool it, then we add a cotton plug into a fermented and allow to a fermented at a room temperature, that is your 37 degrees Celsius for a 24 hours. Okay, so this fermentation will be take place at a room temperature. Then we do a bottling. This is your probiotic beverages. Okay, so what is this probiotics? This topic will be discussed in a next lecture in a detail, because this is very vast, like what is the probiotic drinks and probiotic beverages. So this is all about the lactic acid fermentation. I hope you will understand. For a further query, you can follow the ITLS Academy on different platform like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. Or for a further query, you can ask your query to the helpline number that are already mentioned in your slide. That is 7080833450, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.